We're going to talk about some of the history of microbiology, and I'm going to tell you most of the key stuff here. So back in the day, there was this belief in something called spontaneous generation. We know this isn't true. This was the belief that microorganisms just came out of nowhere. Superstitious kind of stuff. Oh, if you're a bad person, you're going to get tuberculosis. It's not an infection. It's not caused by an organism. It's just because you're bad. Like things like that. So that's what spontaneous generation belief. Today we have this theory of biogenesis, which states life can't just come out of nowhere. It has to come from other life. So cells have to reproduce and divide and form other cells. And the person who came up with the theory of biogenesis, his name was Louis Pasteur, which we'll talk about. Pasteur, you can imagine pasteurization named after Louis Pasteur. Okay, so the founding fathers of microbiology. The first one you need to know, Antony van Leeuwenhoek. So this guy had an interesting story. He was a linen merchant. So he was like this dude who made clothes. Like back then they had like sewing and stuff like that. So he was a linen merchant, and he made this little I'll show you. He made this little device. This is the actual picture, because he wanted to look at the thread when he was doing his sewing, and somehow he actually started seeing these little critters. And we know now that they're protozoa or maybe bacteria. So he inadvertently made the first microscope. These are actually his actual drawings of what he saw. And I'm going to write this down. He called these things that he saw, he called them animalcules. So we credit him with the first microscope, and he saw these little critters. All right, our next scientist is this dude named Edward Jenner. Edward Jenner discovered the first vaccine. By the way, this first vaccine was for smallpox. And just so you guys know, smallpox is the only disease completely eradicated. We haven't had a case of smallpox since 1974. It's gone, thanks to this vaccine. And because of that, we don't get the smallpox vaccine anymore. But your parents or your grandparents might have got it, and they used these metal prongs in the arm. So if your mom or your grandparents or your parents have like a little scar, it's from the smallpox vaccine. But we don't get it anymore. All right, so let me tell you this story. So back in the day, there were these milkmaid women, and they milked the cows. This guy, Edward Jenner, he noticed that these milkmaids would get these little pocks on their hands from the cows, cowpox. Then they would never get infected with smallpox. Like the milkmaids were totally good. They never got it. So he had this idea. He said, what if I take some of the goo from your cowpox on your hands, and I put it into this little boy? He grabbed his own son. This was crazy back then. He grabbed literally his own son and injected his son with the cowpox. Then he waited like I don't know, a couple weeks, and then he injected his son with smallpox, and his son was fine. He survived. So that was the invention of the first vaccine. And then from there, he went on and did a whole bunch of other people, and they found, they had immunity to it. So yeah, that's the first vaccine, Edward Jenner. Next person you really want to know about, this guy, Joseph Lister. Hey, look at this name. This is how I remember this. Guess what they named after Joseph Lister? I'll tell you. Okay, Listerine. Joseph Lister, he discovered this thing called aseptic technique. I'm going to write this down. It's really important. Aseptic technique. Now, any technique you use in the medical field, in your house, in the lab, any technique you use to prevent contamination. Okay, so it's used to prevent contamination of microorganisms in your whatever you're working with. Can you think of an example in the medical field of something you would do, a practice you would do to keep microorganisms away? A lot of sanitizing. You were going to use formaldehyde to preserve. Formaldehyde is the one they use a lot. Remember when you dissected animals, they preserved those animals in formaldehyde because formaldehyde is an antimicrobial. Whenever you go to get blood drawn, what do? They wipe your arm with one of those sanitizing wipes. That's an antiseptic. Guess what else they do? They use a sterile needle every time. Use a fresh, sterile needle to keep any contamination out of your bloodstream. That would be terrible, right? When you're implanting a catheter, a urinary catheter, you're going to be using sterilized tubing. You're going to be wiping with alcohol. All these things are aseptic techniques. If you've ever used Listerine, remember how your mouth burned and it felt really clean? That's what aseptic technique, because it cleans and sterilizes, gets rid of all those microbes, right? So Lister, aseptic. All right, this next dude, Louis Pasteur, did a lot of stuff. This guy was just like experimenting. He took this flask. It's called a swan neck flask because it had a swan neck. And he filled it up with a broth. Broth, you know, like with chicken broth, like you eat when you're sick. Broth is any liquid that has a lot of nutrients. Bacteria love broth. It's like their food, okay? So he took broth and he boiled it to kill all the microbes. And he noticed that when he just left it up, this swan neck flask, he left it up, all the bacteria gathered in the neck of the flask. And he was like, wow, so bacteria don't just come out of nowhere, because none of the bacteria got into the broth. He's like, I thought bacteria would just pop up out of nowhere. But then he tilted his swan neck flask to the side, and now broth was touching the neck, and bacteria got in, and they began multiplying and growing. So the purpose of this experiment was he was showing life cannot just evolve out of nowhere. Yes, there's bacteria in the air, but they actually have to come in contact with the broth in order to grow. So that's how he basically came up with this theory of biogenesis. So what you need to know, Louis Pasteur, he disproved spontaneous generation with that swan neck flask. He also developed pasteurization. Pasteurization is the brief heating, usually milk or food, brief heating to kill pathogens. But pasteurization doesn't kill everything. That's why milk goes bad. Like, even if you didn't open the milk, it'd still go bad within a couple weeks, because it doesn't kill everything. It only kills, mostly the pathogens. So Louis Pasteur is one of the more important ones. He did a lot of stuff. These guys, Tyndall and Kahn, they discovered these things. Another thing you're going to say out loud right now, endospores. Those endospores comes up like a billion trillion times. Endospores are heat resistant, dormant forms of bacteria. And usually they're dormant because they're starving or they live in a really bad environment, nutrient poor. But then once they get into a nutrient rich environment, they can become alive again. And what makes endospores so dangerous is that, one, they cause some of the most deadly diseases on the planet. So some examples, tetanus. Tetanus is caused by a bacteria called Clostridium tetani. It makes endospores. Botulism, which is a foodborne endospore illness. Both of tetanus and botulism produce neurotoxins that are very deadly. Anthrax. Okay, now, does anyone work in like a nursing home? Maybe someone does. Definitely heard of this. C. diff. Clostridium difficile. C. diff is an endospore former. So that's why we got to worry about endospores. Like, especially in like a nursing home environment, there's a lot of endospores, and it can be transmitted from one patient's room to another, and then you're spreading endospores, and then the endospores get into the body and they germinate. So yeah, that's the other thing, is that they're common in the hospital environment, especially C. diff. So we're going to talk about endospores a lot this semester, so you definitely want to know what they are now. There's only one way to get rid of all life. I should tell you this word. If you see the word sterile, free of all life, including endospores and viruses. Viruses are not really life, but we'll put that in there as well. So sterile means free of all life. Completely to sterilize something is to get rid of any form of life or viruses or anything. There's only one way, one foolproof method to sterilize something, and that's to use something called an autoclave. So an autoclave, if you've ever worked in a hospital, you've probably seen one. Yeah, there's like these, they're like big pressure cookers. So basically there's very high heat, 121 degrees Celsius, and high pressure, and you usually autoclave something for like 30 to 60 minutes. Any type of metal, like like the dental office, when you go to the dentist, I'm always like, oh, did they, like, where did this equipment come from? Did someone, it wasn't in someone else's mouth? No, they did, right? So the autoclave at the dentist, most hospitals have them. A lot of tattoo parlors and piercing parlors now have little autoclaves because they have to autoclave the needles and things like that. And any lab, most research labs will have these. Now, some materials cannot go at really high heat, like certain plastics might melt in the autoclave. So there are some chemicals and certain gases that also can sterilize, but only for like certain materials that can't be at that high temperature. Okay, so Tyndall and Kahn, they discovered endospores and they discovered sterilization. Robert Koch, he discovered that specific pathogens cause specific diseases. Only Vibrio cholera causes the disease cholera. Only influenza causes the flu. Okay, so he discovered anthrax. Only Bacillus anthracis causes anthrax. Only Mycobacterium tuberculosis causes TB. So he identified that specific organisms cause specific diseases. And he developed these things called Koch's postulates. And we'll talk more about that later. He also developed pure culture methods. That means he figured out ways to grow specific strains in the lab, just one culture. We'll talk about that later. Let's make a table.